Okay, welcome back to chapter 15. We are going to talk about linked genes. Linked genes are genes that seem to be inherited together. So when you have two different traits, instead of them independently assorting out, you just see them kind of tend to show up in populations together. A linked gene they're going to show up together because they're on the same chromosome. So each chromosome that you have has hundreds or thousands of genes, except for the Y chromosome. They all have hundreds or thousands of genes. Genes that are located on the same chromosome tend to be inherited together and are called linked genes. So Morgan did experiments with fruit flies to see how linkage could affect the inheritance of two characters. So he crossed flies with different body color and different wing size. So the first generation was a P generation, right? So they were all homozygous for these two traits. And then he created an F1 dihybrid flies. So they would all have normal body, gray body, black body, normal wings, vestigial wings. So gray was the normal phenotype, black was a mutant. Wild type flies had this normal large wings. Mutant flies have what's called this vestigial wing. These little almost tried to develop but didn't actually develop wings. So the F1 hybrids should have been hybrid for everything, and then made a test cross. He made a test cross by taking his F1 dihybrids and crossed them with a homozygous recessive. So same as this first generation. He is homozygous for body color, he is homozygous for having the vestigial wings, and had a test cross for his second generation. What you'd expect to see if these were independently inherited is a normal chance of having any combination, right? A normal type of being gray with normal wings, a normal type of being black with normal wings, an equal chance of being black with vestigial wings, and an equal chance of being gray with vestigial wings. But this is not what he saw. He did not see normal ratios. Instead, what he saw was something closer to 1 to 1 to 0 to 0. So he found that body color and wing size were usually inherited together in specific combinations. They were going to match their parent phenotype, either gray with normal wings or black with vestigial wings. Those two traits showed up together. They didn't really separate from each other. So those two genes did not assort independently. Okay, remember independent assortment, instead of saying normal and wild type, instead of saying normal and mutant, could for each trait independently go to any gamete. Those always tend to stay together. And he figured that they did that because they were on the same chromosome. So if a female had two different parents, one gave her the normal body color and normal wings, and one gave her mutant body color and mutant wings, they were on the same chromosome, so they are going to be inherited together. So when they pass on to the next generation, she's either passing these two together or these two together. She's not going to pass on combinations of those. Sometimes non-parental phenotypes did show up. So what was happening in these non-parental phenotypes is these were breaking right here and making new combinations. So you would have to actually break this to combine the normal body color with the mutant wing, or the normal body or the mutant body color with the normal wing. You'd have to break them and swap them around. So the reason we sometimes saw non-parental phenotypes was because of genetic recombination. So genetic recombination relates back to what we talked about with Mendel and Morgan, based on the chromosomal basis of recombination. So offspring with a phenotype matching one of their parental phenotypes are called parental types. Offspring with non-parental phenotypes with a new combination are called recombination types or recombinants. So our parental types were wild type body color, wild type wing, and mutant color, mutant wing. Those are our parental phenotypes. Recombination types would be normal body color, mutant wing, or mutant body color, normal wing, right? Those, you don't see those combinations. In our our peas, we would see either yellow seeds that were smooth or green seeds that were wrinkled. So these are your two parental types, yellow that are smooth, green that are wrinkled. In the offspring, though, we also saw yellow that were wrinkled and green that were smooth. These are recombinant offspring, okay? So these have sorted out differently than what the grandparents were like. These sorted out to be the same as what the grandparents were like. Okay, although some genes were linked in in Morgan's work, some genes were linked, but non-parental allele combinations were still produced. So he said there was some process that occasionally broke the physical connection between genes on the same chromosome. So instead of having these parental phenotypes, they broke. And so this section is now a recombinant, and this section is going to give you recombinant traits. Whereas this one will give you a parental phenotype, this will give you a parental phenotype, this will give you a recombinant phenotype, and this will give you a recombinant phenotype. So the mechanism of having recombinant phenotypes was due to recrossing over of homologous chromosomes. So let's watch this video.
During prophase 1 of meiosis, homologous chromosomes pair up very closely, and corresponding parts of two non-sister chromatids may trade places. This process of crossing over creates variation by producing chromosomes that combine the genes inherited from two parents. Here the process produced a total of 4 genetically different gametes. There are many ways crossing over can occur. In humans, crossover events happen an average of 2 or 3 times per chromosome pair, greatly increasing the variation among eggs and sperm. Note that crossing over produces some parental gametes with chromosomes like those of the parents, and some recombinant gametes with a mixture of genes from both sets of chromosomes.
Okay. So hopefully this concept makes sense to you, crossing over. So recombinant chromosomes bring alleles together in new combinations and gametes. Random fertilization increases even further the number of variant combinations that can be produced. And the abundance of genetic variation is the raw material upon which natural selection works. Okay, Alfred Sturtevant was one of Morgan's students, and he created what was called a genetic map. A genetic map is when he lists the locations of different genes along a particular chromosome. And he predicted that genes that are almost always, the less that they are, the less recombination you see between them, the closer they are. And the more recombination means that they're further apart. They're more likely to be further apart. So the further apart two genes are, the higher the probability that a crossover can happen somewhere between them. So there's a higher recombination frequency. So here, this is a linkage map. A linkage map is a map that a map of a chromosome based on recombination frequencies. So if you say between gene B and gene CN, there's a 9% recombination frequency. Between CN and VG, there's a 9.5% recombination frequency. And between B and VG, there's a 17% recombination frequency. This bigger number tells you that B and VG must be further apart from each other than B and CN, or that CN and VG are, okay? So distances between genes can be expressed as a map unit, and a map unit represents 1% recombination frequency. Important to note, though, map units indicate relative distance and order. It doesn't give you exact location, so you can't say because of this recombination frequency, I know this gene is exactly here and that gene is exactly there. You just can say this is the order that they must be in, and approximately how close they're going to be to each other compared to the other genes. Genes that are far apart on the same chromosome can have a recombination frequency near 50%. 50% though is that max out. If they were on separate chromosomes, they'd have a 50% recombination frequency, if you remember that clip we just saw. So 50% is the max level that you can have. So here you can see the chance of these two genes having long aristae here and having red eyes, how frequently those are together, having red eyes and a gray body and red eyes with normal wings, normal wings, and normal wings and red eyes. So those are the wild type phenotypes compared to our mutants. They have short iris stay, maroon eye mutants, black body, cinnabar eyes, the cinnabar eye color, vestigial wings, curved wings, and brown eyes. So you can just see in every generation how likely they are to show up together and how likely they are to be separated from each other. So genes that are far apart, so if we're looking at the gene for iris stay size and for red versus brown eyes, they are far apart. So even though they are physically linked on the same chromosome, they behave like they're genetically unlinked because there's so much room, all of this room for a recombination event to happen between them. So these two genes are going to act the same as they would act as if they were on different chromosomes, because the chance of recombination happening is the same as the chance of different chromosomes going to different gamete cells. So Sturtevant used recombination frequencies to make linkage maps of fruit fly genes, and they found that genes clustered into 4 groups of linked genes or linkage groups, and conveniently those 4 linkage groups corresponded to the 4 different chromosomes that they had. So those linkage maps, along with having 4 chromosomes, provided evidence that those chromosomes, those genes were located on those 4 chromosomes. They didn't actually know at that point that they were having these different genes on chromosomes, or if those genes were on something else. But it kind of suggested, because there were 4 linkage groups and there were 4 chromosomes, that they probably somehow were related to each other. Let's talk about some genetic disorders. So all the things we've talked about over the last few chapters are kind of the typical what you expect to happen, but alterations can happen. Big chromosomal alterations in humans and other mammals often lead to miscarriages. Miscarriages are also called spontaneous abortions. So a baby just won't be able, a fetus won't be able to develop to birth because of this, if they have certain large-scale chromosomal alterations. Some babies are born with some chromosomal alterations, and they may or will likely have a variety of developmental disorders. Plants, however, are able to tolerate a lot of genetic changes better than animals do. We have a lot of plants, a lot of species based on the genetic changes that they've tolerated and thrived even with. Okay, so some different ways that we have some different situations in our cells. One thing that can go wrong is nondisjunction, and nondisjunction is going to give you abnormal chromosome numbers. In nondisjunction, instead of having every homologous chromosomes going separate ways, so you have sister chromatids all in individual cells, and then in meiosis 2 go on and have normal cells. Non-disjunction means that they don't separate, they don't disjoin. So here, if you have non-disjunction in meiosis 1, you're going to end up with all your cells either have an extra chromosome or missing a chromosome. A non-disjunction in meiosis 1, all of your cells, they either have an extra chromosome or missing a chromosome. If non-disjunction happens in meiosis 2, so this cell is going to go on to create 2 normal gametes, but this is going to go on to create half of your gametes have an incorrect number of chromosomes. So 1 out of 4 will have an extra chromosome, 1 out of 4 will have missing a chromosome, and half of your chromosomes will have, half of your gametes will have normal chromosome number. Okay, so non-disjunction, whether it happens in meiosis 1 or meiosis 2, if your chromosomes don't properly separate during meiosis, then you end up with extra chromosomes or too few chromosomes. So here's meiosis, here's non-disjunction. Oh, where did it go? Try it again.
See what happens when mitosis malfunctions. Newt epithelial cells were grown on cover slips and photographed using differential interference contrast microscopy. Notice how in this cell, one chromosome, black arrow, never arrives at the equatorial plate, likely due to improper attachment to the spindle fiber. As in meiotic non-disjunction, This will result in the daughter cells having abnormal numbers of chromosomes. Credit: Conley L. Ryder, Wadsworth Center, New York.
Okay, so nondisjunction, even though we just saw it happening in mitosis, it can happen in mitosis. We're typically going to talk about it in meiosis more because that single gamete is going to go on to develop a whole body, a person who all their cells have either an extra chromosome or missing a chromosome and are more likely to have a deficiency, whereas this is just going to have, a couple of cells that are going to, whatever cell is being created, just a couple of cells, not the whole organism itself being affected. Okay. Aneuploidy is when you have gametes fertilized that have after you have nondisjunction, if that gamete goes on to be fertilized, that gamete, that zygote is going to be aneuploidy. So aneuploidy creates offspring with an abnormal number of a chromosome. If you have an extra chromosome or if you have a missing chromosome, either way, that is aneuploidy. A monosomic zygote has one copy, so mono for one has one copy of a chromosome when they should have two, and a trisomic zygote has 3 copies of a chromosome when they should have 2. So you may have heard of some diseases like trisomy 13 or trisomy 18 or trisomy 21. That means that you have an extra chromosome, and because you have an extra chromosome in the zygote, as that baby develops, all the cells in their body will have that extra chromosome. Polyploidy is a condition when an organism has more than 2 complete sets of chromosomes. So polyploidy, anything more than 2. Trisomy, so triploidy, when you have 3 sets of chromosomes. Tetraploidy, when you have 4 sets of chromosomes. You have the complete set of your chromosomes. Not only do you have an extra copy of one chromosome, you have an extra copy of all the chromosomes. Polyploidy is pretty common in plants and has actually led to a lot of variation in plants that we see now. It's not very common in animals. And polyploids are going to be more normal in appearance than aneuploids. Okay, let's talk about some other ways that chromosomes can be altered. So if a chromosome breaks, this can lead to 4 different types of changes. One is a deletion. Deletion is when you remove a fragment. So here you have genes A, B, C, D, E, F, G, H, and it's going to break before and after gene D. So now you have A, B, C, E, F, G, H. One is a duplication, when a segment gets repeated. So here you have A, B, C, D, E, F, G, H. Here you have A, B, C, B, C, D, E, F, G, H. So B, C has been duplicated. An inversion is when a section breaks off and flips back in backwards. So instead of having A, B, C, D, E, F, G, H, you have A, D, C, B, E, F, G, H. And sometimes these genes will be able to function normally, and sometimes those genes are not going to be created normally. They're not going to be expressed normally at all. And then a translocation moves a segment from one chromosome to another. So here you have this chromosome and this chromosome, A, B, C, D, E, F, G, H, and then a non-homologous chromosome that is MNOPQR. And you break off. So now you have this chromosome and this chromosome, MNO, C, D, E, F, G, H, and ABPQR. Sometimes that will still let these genes be expressed normally. Sometimes that can have massive implications for how that infant develops. And then if that, in meiosis, you end up with cells, you don't quite have the correct homologous chromosome, so you end up with a lot of issues in that next generation. Okay. Alterations of chromosome number and alterations of chromosome structure can be associated with some serious disorders. Some types of aneuploidy appear to upset the genetic balance less than others. And so aneuploid individuals are often able to survive to birth and beyond, although they may have a certain set of symptoms or syndrome characteristic with that type of aneuploidy. So let's talk about trisomy 21, or Down syndrome. Trisomy tells you have 3 copies of chromosome 21, see right here. So instead of having 2 copies, you now have 3. This affects about every 1 out of 830 children born in the United States. And the frequency of Down syndrome increases with the age of the mother, and maybe even with the age of the father, although we don't really know why at this point. A mother of any age can have a child with Down syndrome, but the chances of it increase as age increases. We can also have aneuploidy of sex chromosomes, and that is because the sex chromosomes had nondisjunction and ended up with a variety of aneuploid. One is called Klinefelter syndrome. Klinefelter syndrome is when a male has 2 X chromosomes and 1 Y chromosome. Typically, these males will have a slightly more feminized in some traits, but still be able to develop. About 1 in 1,000 men have XYY. They have an extra Y chromosome. And these males don't really have any syndrome at all. They just have an extra Y chromosome there. XXX females, these are females that have 3 X chromosomes. This is about 1 in 1,000. They are healthy, no unusual physical features, although there's some risk of learning disabilities. And then monosomy X. Monosomy X means you have 1 copy of your X chromosome. So you'll become a female. This is called Turner syndrome. So you'll have 1 X chromosome and the other chromosome you only got 1 sex chromosome. Individuals with Turner syndrome are typically sterile. And interestingly, this is the only known viable monosomy in humans. So there is no monosomy 1 or 2 or 3 or any of them. The only monosomy in humans that are viable, that survive to birth, are monosomy X. There's a syndrome called cri du chat, which means cry of the cat, because these babies cry typically sounds like a cat mewing. This is a deletion in chromosome 5. And children born with cri du chat are severely intellectually disabled. They have a cat-like cry, and they usually die in infancy or early childhood. Certain cancers, including chronic myelogenous leukemia or CML, are caused by translocations of chromosomes. So here, this tip of chromosome 9, tip of chromosome 22 separate, and this translocated chromosome 22 is known as Philadelphia chromosome. Okay, there are some inheritance patterns that are exceptions, normally occurring exceptions to Mendelian genetics. One exception is genes located in the nucleus, and one exception is genes outside of the nucleus. So you do have some genes, particularly your mitochondria have genes that are outside of the nucleus. They just stay in the mitochondria. In both cases, the sex of the parent contributing an allele is a factor in the pattern of disease. So it matters if your dad or your mom gave you the bad chromosome, the defective chromosome. Okay, so this is because of genomic imprinting. For a few traits, for a few mammalian traits, the phenotype that you develop depends on which parent passed you the allele for those traits. Genomic imprinting involves silencing certain genes depending on which parent passed them on. So if the mother or the father passed on the gene would depend on what your phenotype is actually going to be like. So here, this wild type mouse has a normal allele and a defective allele. The maternal chromosome, the gene is not being expressed, so they are expressing the normal allele. They are normal. That is, you can have heterozygotes that have a normal LF2 gene inherited and a mutant inherited, but they silence the mutant, so they are normal. If they have the mutant from their dad, though, they silence the normal one. They express the mutant allele from their dad. They're going to be mutant. Does that make sense? So inheriting it from their mom, they don't express that. They're normal. Inheriting it from their dad, they do express it. They have that dwarfism. That is genomic imprinting, when silencing a gene depends on which parent passed it to you. Imprinting, we are still kind of figuring out what's going on there, but it is likely because you methylate. If you remember methylation, methyl is one of those functional groups. You put a CH3 group on certain nucleotides. Genomic imprinting probably only affects a small fraction of our genes, and most imprinted genes are critical for embryonic development. Extranuclear genes or cytoplasmic genes are found in the organelles. So mitochondria, chloroplasts, and other plastids carry small circular DNA molecules. And extranuclear genes are inherited maternally. because sperm do not carry organelles, only the egg have organelles. So all the organelles you have came from your mom. So your mitochondrial DNA is all from your mom. Genetically, it's a nice way to trace your maternal lineage. First evidence of extranuclear genes came from studies on the inheritance of yellow or white patches on leaves of an otherwise green plant. A lot of how plants' coloration develops is based on those mitochondrial DNA or chloroplast or plastid DNA that are affected from their mother. Okay. Some defects in mitochondrial genes can prevent cells from making ATP and can give you diseases that affect the muscular and nervous systems because they need a lot of ATP. So mitochondrial myopathy and Leber's hereditary optic neuropathy. These are diseases from defects in mitochondrial genes. So a father can't pass them on to his children, but a mother always will. It could be possible to avoid passing along mitochondrial disorders if you take the chromosomes from the egg of an affected mother and put them into the egg of a healthy donor. So you have an egg from a healthy donor, take out its chromosomes, put in the mom's chromosomes, the mom who wants to be the mom, and then fertilize it with sperm from the father, and then transplanted that back into the prospective mother. So something similar to IVF, where you use two different mothers, you use eggs from two different women. So the children are going to grow up with the egg donor's mitochondrial DNA, but their genetic mother's nuclear DNA. Okay, so I'm going to let you read these last few slides on your own. And as always, you can contact me when you have any questions, and I'm happy to talk anytime you need. I'll see you later.